Hello and welcome, everyone. My name is Dennis Koester, and I'm your host today for today's Drug Hunter Flash Talk. We will be talking about bioisosteres with Nick Meanwell today. So welcome, Nick. We're really glad to have you. It's a pleasure to being able to host you.
Well, and thank you for the opportunity, Dennis. I'm very much looking forward to it. Thank you.
Awesome. So without further ado, I want to introduce our guest, Nick. And Nick doesn't, of course, need much introduction, as probably everybody knows him. Nick had more than 40 years of experience in drug discovery and led drug discovery projects in a variety of different therapeutic areas, such as cardiovascular diseases, neurosciences, and also infectious diseases. And his research resulted in the advancement of 33 clinical candidates for the prevention of thrombosis, the treatment of stroke, and therapy of viral infections, including HIV, HCV, and also RSV. And Nick is, as you know, a very prolific author and has authored or co-authored more than 300 publications, review articles, book chapters, and editorials, and more than 200 meeting abstracts as well. And he's named the inventor or co-inventor on 144 issued US patents, so really prolific. Nick, of course, received a variety of different awards, many awards during his career, but he was also inducted in the MEDI-ACS Hall of Fame and received the prestigious Hero of Chemistry Award not only once, but twice. So yeah, and those are, again, just a few awards that he has received. Nick obtained his Ph.D. from the University of Sheffield and started his career and his industry career at BMS in 1982 after a postdoc at Wayne State University. And Nick formally retired from his position as senior vice president at BMS in 2022. Our team actually visited a couple of weeks ago, and Nick is dearly missed at BMS. But thankfully for all of us, Nick is still very active in the medicinal chemistry community. So we still all really benefit from his knowledge, and he cares deeply about the education of the community. So with that, Nick, it's a pleasure of having you today. So yeah, thank you for taking this opportunity, being here with us today, and talking about bioisosteres.
You're very welcome. As I say, I'm very much looking forward to it. It's one of my favorite topics.
Awesome, awesome. Yeah, we like to get these started with some warm-up questions. We're going to have a shortened version today. So, before we start, I wanted to ask you, Nick, what's your favorite molecule and why?
Thanks, Dennis. So that's an interesting question. And my favorite molecule, I obviously have several. We've gone into the clinic, and obviously decladazole is one of my favorites because of the process by which we discovered it was fascinating. But really, my favorite molecule is another BMS compound, apixaban, antithrombotic factor 10A inhibitor from Don Pinto's group. I needed to take it about 3 years ago when I had a deep vein thrombosis, and I remember taking that drug, and within 4 to 5 hours, my symptoms were starting to lift. So it was a life-saving thing, an event. In its essence, I had multiple pulmonary embolisms that could have been fatal or even caused a stroke. So and I was really touched by that. An important compound,
Wow,
and
yeah,
one that I specified actually because of its profile.
no, I mean, that's a life-changing effect. I mean, that medications can have. So I love these personal stories. Thanks for sharing that with us, Nick. All right, one more question before we kind of get started. What are you most excited about in drug discovery today?
I retired, but the opportunity in front of drug discovery is just immense. So part of me has sort of disappointment in that sense because when you look at the current modalities, the way we're screening, the target ID, the tools that we have today, I just see an opportunity in front of small molecule drug discovery that is bigger and broader than anything I saw in 40 years. And whilst my generation and the generation before did, I think, wonderful things with the tools we had, the tools that are available today are so much more superior that one can anticipate, I hope, a continuous flow of really important drugs PROTAC being a classic example of a technology that has the potential to be transformational. I'm sure it will have a few hiccups on the way to its successes, just like everything else does. But that and what it spawned with proximity-induced effect is remarkable.
Yeah, 100% can agree with that. I mean, so many different modalities to be excited about. And I think few of the molecules we will see today in your talk, which will be very exciting. Before you pull up your slides, I briefly wanted to share a feature with the audience here on Drug Hunter that you can use to find bioisosteres, and that is the structure search here. So if you head over to Drug Hunter, sign in and go to that structure search, one of the things you do, for example, looking for different isosteres, let's say you want to know how many drugs are approved that have a deuterium atom. So you can just draw deuterium here, head over to drugs, search for structure, and you will find quickly the drugs that are approved that contain a deuterium atom. Here, most prominently, of course, the deucravacitinib, which was one of the molecules of the year at Drug Hunter. It was approved in 2022. And also, of course, since the set of FDA-approved drugs is somewhat limited, if you want to search for really interesting preclinical compounds or clinical candidates, you can also search our molecule database. So we just, in the discussion with Nick, I talked a little bit about fluorine. So, for example, if you're interested in a trifluoromethoxy group or, anything like that, and Nick will talk a lot about different fluorine bioisosteres during his presentation, just draw that structural motif here in the box and then click search for structure. And you'll be able to find a number of interesting clinical candidates that are sorted out for you. So, the advantage over things like SciFinder really is that, you find relevant medicinal chemistry results, drug discovery campaigns, not results, the hundreds of thousands of results that you find on SciFinder, how to make trifluoromethoxy groups, but really what was the impact on the properties? Why did they use this particular group? And it's also really filtered for drug discovery. On other databases, you would find things like from material science, of more academic interest, but here you can really find a large variety, and you will learn why those groups have been employed and whether or not you should be trying those on your own project. And with that, without further ado, I'll hand it over to Nick for his presentation. And that'll be entitled The Chemist's Playbook. We really have the tools, as Nick said, on our fingertips today to change the way drug discovery is done. And we'll learn more about this from Nick now. Stage is yours.
Thank you, Dennis. Let me just get my pointer going. So I'm going to give you a little bit of a primer, if you like, on impactful bioisosteres. This is a huge subject, and it's not easy to choose representative examples, but I'm trying to give you a flavor of how bioisosteres have actually had a big impact on drug discovery, drug development, and marketed drugs, actually. This is an outline of the talk. I'll give a little bit of background. I'm going to focus on a few isosteres I've selected, some from our own work, carboxylic acid bioisosteres, some amide bioisosteres, obviously replacing hydrogen atoms, and Dennis has just discussed fluorine and deuterium. I think these are important bioisosteres, as you'll see. And then I'll end up with a slide on phosphate isosteres, particularly the nucleoside phosphonates that have been transformational in HIV, and then bicyclic pentanes. And I'll have a little bit, I've got a slide on emerging bioisosteres we'll discuss at the very end before the conclusion. If you look at why we use bioisosteres, there are manyfold. Potency and selectivity, we'll see examples of that, conformational effects, physical property modulation, and improving drug-like properties. It can address, reduce, redirect metabolism to eliminate or modify a toxicophore, an undesirable element in a molecule. And it's oftentimes used for novelty as well. But the applications of bioisosteres is contextual. So you really have to tailor and select a particular bioisostere for a specific application, because what works against one target may not work against another target. So the context is important. And as you see, bioisosterism started out as isosterism, similar shape, and it's now evolved into contemporary usage where the molecules can be quite complex in structure and really not at all of the similar shape or even electronic characteristics, but there's clearly something in these motifs that can emulate another functionality. So they're often more alike in biological rather than physical properties, and what I call higher order bioisosteres, where they are structurally really quite complex molecular metaphors that takes the concept well beyond that of just a simple shape replacement. This review here by Thornberg, I discovered that when I first started BMS, and I had to print that out multiple times because I dog-eared it to the point where it needed to be replaced. And this one by Alfred Berger is just fantastic. This captures everything up to 1991. It's both comprehensive and in depth. If you look at the origins, the isostere concept was from 1919 by Langmuir in this Jack's paper, which you can still go back and read, called Isomorphism, Isosterism, and Covalences. And he was anticipating that molecules with similar arrangements of topology would have similar physical properties. And so he coined this term isostere. He gave 21 examples, some of which are shown here. Most interestingly, he predicted that ketene would behave a lot like diazomethane, which was known at the time. Ketene wasn't synthesized until 18 years later, when this was then confirmed. Probably one of the first examples of a bioisostere in a biological setting came out from the work of Karl Landsteiner and Hans Erlenmeyer. This is not Erlenmeyer v. Erlenmeyer Flask, a different one. They were making artificial antigens by reacting diazonium ions with proteins. So these would specifically, as you can see here, modify the ortho position of a tyrosine. So this gave them an opportunity to vary, obviously, this phenyl ring here, this substituent. So Erlenmeyer was developing these antibodies, and he showed that the antibodies could not distinguish, for example, between these three motifs, where you've replaced a CH2 with an oxygen or an aniline, and couldn't distinguish between a phenyl ring and thiophene. So the first example of what now is a sort of classic bioisoster, thiophene for a phenyl. The term bioisostere was not introduced, though, for another 20 years when Harris Friedman introduced it in 1951 to define compounds that fit the broadest definition of isosteres and have the same type of biological activity. So these are the classical isosteres, pretty straightforward. We'll look at deuterium and fluorine for hydrogen and some of these others, and the bivalent, trivalent, and tetravalent. And of course, the ring equivalents we just talked about here again, thiophene for benzene. The more interesting ones are the more contemporary versions that Ed Lavoie, again, this really nice review in 1996, talked about non-classical bioisosteres. He divided them into exchangeable groups and compounds with rings versus non-cyclic structures. So here his examples were changing an ester or an amide into an oxazole, an azole of some sort, work I think that was pioneered by John Saunders at Merck back in the 1980s in the context of the Ricoline. Exchangeable groups here, replacing a phenol with a methyl sulfonamide. That was work from Ollie Larson, who was actually conducting most of his work at the site that I started my career in Evansville, Indiana. And the only similarity here is the pK of that proton. But you can see that's a much more polar element, and it's actually quite large. But there are places where that works beautifully as a phenoemetic with certain advantage. Ring versus non-cyclic. This is an interesting topology. It was explored again in the beta blocker area to replace a phenyl ring with something a bit simpler. And we'll look at this, the acid tetrazole bioisosterism, where you can replace an acid with an acidic azole. And then there's this shape mimetic, where a group at Novartis did a nice piece of work on BCL inhibitors, looking at this self-association at the end of the molecule and replacing that with this three-dimensional adamantane. And as you look at non-classical ones, Chalcogen bonding, shown here with chlorine, bromine, and iodine, but not fluorine. They don't have low-lying sigma star orbitals. This is an area of positivity. That is essentially the CX sigma star orbital, and there's two of them on sulfur. It's a CS bond. These are looking for electrons, areas of electropositivity. So are hydrogen bond donors. So these are essentially equivalent to NH and OH, and there are examples of that. There are two on sulfur. These are usually intramolecular because of the topology, whereas Halogen bonding can be intermolecular. You can deepen and asymmetrize those bonds by converting the thioethene to a thiazole. Does not exist on simple alkyl sulfides. You need an electron withdrawing group on that C alpha. And it also works in with this interesting difluorochloromethyl moiety. Difluorochloromethyl is not the same as trifluoromethyl. Very different. Probably one of the first examples, long before the term bioisostere was coined, is in the sulfonamide antibacterials discovered in the early part of the last century. Sulfanilamide, shown up here, was discovered in 1908 and used in World War I. Prontazil is a prodrug. This NN bond gets cleaved to release the sulfonamide. These are antibacterials. These actually, it turns out, mimic para-aminobenzoic acid. So here, in this case, the sulfonamide is mimicking a carboxylic acid, although it's nowhere near as acidic. But you can acidify it by introducing electron withdrawing groups onto that nitrogen. So here's sulfapyridine, MMB 693, the first compound to develop, discovered in 1937 and approved in 1938. And there have been now a whole range of these. And here's the X-ray crystal structure data taken from this paper of sulfathiazole bound into the active site. And you can see these sulfones and the NH are important for drug target interactions. So an early example of a bioisostere before we even appreciated bioisosteres. Looking more at acids, and again, acyl sulfonamides, which is what those compounds on the previous slide were. This is one of our contributions to the HCV problem. Looking at HCV NS3 protease inhibitors. This is a really difficult target for the industry. It's a serine protease that has a very flat, shallow active site. Excuse me. And for which carboxylic acids had been identified as inhibitors by Boehringer Ingelheim with some beautiful classical medicinal chemistry. We had wanted to fill this small invagination, the P1 prime pocket. So we needed a way of introducing a substituent here whilst preserving the acidity, because we needed that acidic moiety there. So we conceived of an acyl sulfonamide, and you can see here when we made the methane sulfonamide, we were equivalent to the acid. But the most impressive compound in that series is the cyclopropyl, where we're looking at being about 50-fold more potent both against the enzyme, but also over here against in the replicon cell system. And if you look at the X-ray crystal structure that we solve, you can see the sulfonamide is engaging both the catalytic serine, but also the oxyanion hole, the NH of glycine, which is also engaging the carbonyl group of the acyl sulfonamide. It's a very unique shape. Just adding 2 hydrogen atoms to that cyclopropyl will reduce potency by 20-fold. It just widens that moiety to a size where it's just that little bit too big. So I use this as an example of Julius Riebeck's 55% pocket filling rule, and you can see the difference there is quite profound. And this motif now is in all the marketed second generation compounds, isonaprovir. Acid isosteres were critically important in the angiotensin II receptor antagonist area. The non-peptide ones that were pioneered by DuPont Merck, my former colleagues Dave Carini and John Duncia. And you can see here's the prototype compound. It's carboxylic acid, 200 nanomolar. John Duncia contributed the tetrazole that improved potency by 10-fold, big difference. And this molecule actually is losartan, shown over here. It's a prodrug in vivo. It gets converted to the diacid. The way they rationalized this was the distance. This is 2.2 angstroms. With the tetrazole, you actually get half a bond further projection of the acidic NH if you go out to this position. It's a little bit longer here, but this allows it now to engage more effectively with an arginine 167, which is in the extracellular loop of that receptor, based on this crystal structure data published almost a decade ago. And again, as you think about this acyl sulfonamide also is quite effective. It's a little better than the acid. But again, if you flip the topology of that and put the sulfur onto the phenyl ring, the potency increases quite significantly, because again, that longer carbon sulfur bond, sulfur nitrogen, pushes this acidity NH or the anion further out, and because of the potential for delocalization on this oxygen, you've actually got even further reach. So again, that was in losartan, the first marketed A2 antagonist. For amide isosteres, again, I'm drawing here upon some of our work in HCV NS5A. This was a bit of a breakthrough compound for us. We were able to get genotype 1A activity actually for the first time. Genotype 1B was always sensitive to these molecules. But this molecule, the issue here was changing an alcohol here for an amine, and that gave us about 1,000-fold increase in potency for a modest increase in molecular weight. You can see this is a linear motif. We've got this aniline embedded in here in this sort of peptide-like element on the end. We were, of course, concerned about the potential for aniline release, and so the way we solved that was to wrap this around into the phenyl ring like this, into a benzimidazole at each end, and we're a little bit less potent, but then now the aniline is embedded in a way that it can't be released in vivo anywhere near as effectively. But we took a 20-fold hit in genotype 1A activity from 30 and out to 260. We solve this problem ultimately by removing the alkyne, which is another potential liability, and that of course shortens the molecule to a biphenyl. But in order to preserve the distance between these termini, which is the pharmacophore, the two ends of this molecule, the pharmacophore, we deanylated this ring. So this now became the core to the molecule. So we've removed the alkyne, we've got a biphenyl, we've restored the cap separation by the deanylation. So this again is an amide isostere now with a slightly different topology. Now this is decladisvir, the final molecule we took to the clinic, shown here, and that was approved. Here's the potency, it's picomolar. Interestingly, we calculated at the EC50 when you've got about 4 or 5 picomolar in a genotype 1B replicon. Decladisvir is outnumbered by about 10,000-fold for its target, which is NS5A dimer. So substoichiometric modulation of viral target. A fascinating example of that. And there are other examples, not quite as profound as this in the antiviral field. This is Arun Ghosh's work looking at amide and ultimately sulfone isosteres. This is sequinavir, Roche's HIV protease inhibitor, and the drug target interactions. It's quite potent. It's got an IC50 of 230 picomolar, but it's poorly bioavailable because it's very peptidic in nature. Ghosh replaced this amide with a tetrahydrofuran, which gets rid of two hydrogen bond donors. Potency has gone up by about 4-fold. He locked off this part of the molecule, which is involved in 2 hydrogen bonding interactions, and he's now 3,000-fold less potent, but smaller. He continued to optimize that. He replaced the tetrahydrofuran with this sulfone, and again, he's got good potency here. And combining this with his understanding of the shape of this, he designed this very interesting bicyclofuran moiety, bis-THF, I guess. This compound has a Ki of 16 picomolar, and this is retained in darunavir, which is a 14 picomolar dose. Probably the optimal protease inhibitor. It's got a very good resistance profile. So this is a replacement for the sulfone. You can see here the drug target interactions, and I've drawn down here how Arun Ghosh views this as a sulfone mimic with the 2 oxygen atoms here nicely overlaying. Dennis talked about fluorine. This is a sort of wallpaper slide of some of the properties of fluorine. Fluorine will affect conformation of prolines. If you've got the fluorine cis to this R group here, the C-gamma endo is favored. If it's trans, then the C-gamma exo conformation is favored. This mimics the effect of hydroxylation on proline in collagen, and it's a function of the Ghosh effect, which stabilizes that particular conformation. Fluorines, of course, you can add about half a log in lipophilicity when you put a trifluoromethyl group in. Fluorine can engage in what is oftentimes referred to as hydrogen bonding, whether it's controversial, whether it's real hydrogen bonding. We'll see an example of a multipolar interaction where the electron density on a fluorine can engage a carbonyl group of a backbone. These are actually electropositive, tetral bonding, but you've got to get the geometry just precise. And here's an example where a dipole effect of a fluorine was thought to influence the conformation of the hinge binder in aurora kinase to influence selectivity, actually. You can affect metabolism. These are planar, these are not. They can go either way. They can fluorinated olefins or trifluoroethylamines are useful mimetics of amide isosteres based on the properties. This is dipole mimicry. This is NH mimicry. That's a non-basic amine with a pK similar to an amide and a similar geometry. There's a bunch of other molecular metamorphs. Fluorine is useful to replace a carbonyl group, a nitrile, even an alcohol, as well as a proton. And again, you can replace the necessary nitrogen in pyridines with a fluorine if dipoles are involved. And you can see here the dipole of the difluor is quite a bit better. Fluorination pattern can affect lipophilicity. The vicinal difluoros are more soluble than the geminals because of the dipole is quite a bit higher. And fluorine will also engage with sulfur, and as we'll see shortly, NHs as well to benefit. Here's the example from my former colleague Don Pinto looking at the effects of fluorination alpha to an NH. And you can see when they change that R prime from hydrogen to fluorine, CaCO2 permeability increased. And the same when this is a CF3 out here rather than a methyl group, you see an increase in permeability. More pronounced over here, the factor 10A inhibitors, by the way, more pronounced over here, where again, changing that hydrogen to a fluorine was almost an order of magnitude more permeable. And that was attributed to a fluorine to NH interaction, reducing the solvation of this NH by water. It was also looked at by the Amgen group, and they were looking to interfere with efflux. Don and his group didn't, they just looked at CaCO permeability. The Amgen group looked at PGP. And by engaging this NH intramolecularly, they were able to reduce the efflux ratio and increase the membrane permeability across a membrane, CaCO cell membrane. And they could, here they're using methoxy, which is a strong hydrogen bond acceptor. You can replace that with fluorine, and the fluorine can also be attached to an aromatic ring. So you see this motif a lot in marketed kinase inhibitors, whether it's adjacent to the NH as it's shown here, or working across, in this case, a sulfonamide or an amide, as shown here. An interesting bioisoster of this motif that has not been really exploited very much is in these papers is using a morpholine. This was done in the context of these ASK1 inhibitors. Again, this is replacing a fluorophenyl with something three-dimensional and a little bit more polar. An unexploited motif. The CHF2 has been prominent over the last decade as a hydrogen bond donor. The origin of that was in the agricultural chemistry field. It was an observation made by John Erickson when he was at Monsanto. He saw this intramolecular hydrogen bond here. It was picked up several years later by Frank Nard when he was at IRBM. This is an HCV-NS3 protease of a hexapeptide with 4 carboxylic acids in it and a cysteine at the P1, which is the recognition element. This is problematic, forms disulfides, etc. If you replace that cysteine SH with a methyl group, you're about 20-fold weaker. He recognized the potential of this as a hydrogen bond donor to replace that cysteine and completely restored potency. Here's the poor man's view of how that works. You've got electron-dense fluorines that mimic the lone pairs on the sulfur, and then the hydrogen's a donor. And you can see in the X-ray crystal structure, the hydrogen bond, there's a hydrogen bond interaction with the backbone carbonyl shown over here. Interesting, one of these fluorines is engaging with it looks like it's engaging with the CH of phenylalanine 154. These have also been around. These motifs are again underexploited in drug discovery, but heavily exploited in the agricultural chemistry field. They do some interesting things, and we're sometimes slow to discover what they've done. Even these will intramolecular hydrogen bond. There are 3 of these in the CSD, and you can see this is the topography likely set up by but not only by the intramolecular hydrogen bonding, but also the dipole alignments. And so this has been used in HCV and H3 protease as well. After Frank Nage did that work, we looked at this in the context of these vinyl cyclopropanes. The vinyl cyclopropane is really good. We couldn't replace this with the Frank Nage type motif. The compound was really poor, and the methyl compound was even poorer. But if you reinstalled it back onto this cyclopropane, you fully restored activity. And again, we could see a hydrogen bonding interaction with the backbone carbonyl of leucine 135 here at a distance of 2.7 angstroms. And again, the SAR is consistent with that playing a role, although clearly it can be mimicked by the vinyl group. But this motif is found in the two marketed compounds, glecaprevir from AbbVie and voxilaprevir from Gilead. Now, we talked about the importance of fluorine. This is from Merck. This is in PCSK9. This fluorine here is really important. And the lead molecule was discovered by Adele screen mRNA display. It contained that 4-fluorotryptophan, as far as I recall. With that 4-fluoro there, the potency is 47 nanomolar in this context. If you remove it, you see a 23-fold drop in potency for the change of a single fluorine atom to a hydrogen atom. And it only amounts to 1%, just over 1% of its molecular weight, so it's a huge effect. Merck pointed out that there's interaction between that fluorine and the NH of glycine 370 and then the carbonyl groups of isoleucine 369 and 380. And you can see here it's pointing straight into that pocket. If you look closely at that, though, they didn't describe that. That fluorine is engaging that carbonyl group of valine 380 almost at a perfect Bürgi-Dunitz angle. And the distance is just below the sum of the van der Waals radii, suggesting this is a multipolar interaction that's contributing to that potency. And this is their orally active compound. Bioavailability is just a percent or two, but it's currently in the clinic. A remarkable story. It's well worth reading that. Dennis mentioned deuterium. Deuterium, the first deuterated compound that was approved was deutetrabenazine, and this was designed to slow down metabolism by demethylation. Deuterium is probably the most conservative replacement you could have for a hydrogen. These are the differences in molar volume and bond length, very, very small. The CD bond is between 6 and 10-fold more stable than a CH bond, so you get differential chemical reactivity, the kinetic isotope effect. Ranges from about 1 to 7-fold, depending on the circumstance. Calculations suggest 7 to 10. Can be as high as 60, and there's even one example from Wilfred van der Bonk where it's up around 200. So this will slow metabolism or redirect it. It's going to change the complexion of metabolites. You have to do a full toxicological workup. The first de novo compound approved was the compound that Dennis mentioned, deucravacitinib, shown here from BMS. It's a TYK2 pseudokinase inhibitor. That deuterated methyl group is there to slow metabolism, not because the compound that's produced is less potent, but because the compound that's produced by demethylation is less selective. So a nice example. And then the most recent one was just approved a few months ago. This JAK1/2 kinase inhibitor from Concert, and they've fully deuterated this almost this cyclopentane ring to slow down metabolic modification. Phosphate bioisosteres, nucleoside analogs require phosphorylation to the mono, di, and triphosphate, and that's a substrate for polymerase. I apologize, there's a spelling typo there. This then reacts is incorporated into a growing DNA or RNA chain, depending on what's here. The late Antonian Holy developed phosphonates to replace the phosphate moiety, and this is tenofovir. It has to be marketed as a prodrug, tenofovir diazoprop, which improves membrane permeability or oral bioavailability. But this is a very replacement for that motif, and the reason it's there. Its first step is oftentimes the most phosphorate that alcohol. You can see here with a cyclic alcohol does get phosphorus, but that's by viral specific enzyme, the thymidine kinase. Here, this circumvents that step, and this is indeed converted into the diphosphate derivative, which is a substrate for HIV and HBV polymerase. The aliphenamide prodrug from the late Chris McGuigan, this so-called protype technology, reduces the dose of this molecule from 300 milligrams down to 25, because this directs cleavage to the intracellular environment. So this is a functional mimic of the monophosphate. And then finally, I think this is the final vignettes are on propylene. This is again another interesting motif that sort of sat in the literature for quite some time before it became sort of mainstream. Policiari in Italy looked at it in the early 1990s in the context of glutamate antagonists shown here. This is his prototype. He's taken glutamate, replaced the alkyl chain with the phenyl ring. I can't find the potency of that compound, unfortunately. He replaced the phenyl ring with the propylene moiety, 25 micromolar. He recognized that he'd shortened the distance between the sort of vertices of the ring from 2.8 to 1.7 angstroms and compensated for that by introducing the tetrazole. Didn't work terribly well here. And that was in 1996. About 10, 12 years later, Antonio Stepan, when she was at Pfizer, replaced this fluorophenyl ring with a propylene in avagasostat. This is Bristol Myers Squibb's gamma secretase inhibitor. Here are the X-rays of these two molecules. They're not quite aligned, and I apologize for that in quite the same way, but they do recapitulate the geometry quite effectively. But obviously it's a little bit shorter. Potency is about the same, and if you look at the calculated log D, it's almost a unit lower. Now these are the efficiency metrics that have emerged over the last 15 to 20 years. You can see ligand efficiency has improved a little bit. Ligand lipophilic efficiency is improved by almost 2 units, so that's really good. In this Aztecs version of the LLE, again, there's about a 30% increase in that number. That's all well and good. What about in real terms? Well, if you look over here at the solubility, the kinetic solubility of this molecule compared to avagasostat is increased by about 400-fold, and the thermodynamic solubility by about 10-fold, whilst the membrane permeability is increased by 4-fold. So, real effects of this kind of a change. I think this was what galvanized the community. Pfizer started engaging with Phil Barron to figure out how to make these. The way these compounds are made are from this propylene intermediate, which is very susceptible to strain release modification. Pharmablock, and I think Enamine are doing the same thing, are now making this molecule by flow chemistry at over 1,000 kilograms per month, making these motifs now readily available to the community to use. And you can see here. So here are the publications by year in SciFinder with substructure and biological activity. Since that publication came out, it's increased almost exponentially. And this with synthetic preparation, it's really there's really been a huge amount of work there. And now you're seeing appearing in clinical candidates. This ELF2B activator from Denali contains this motif. It replaces either a diamino cyclohexane or a phenyl ring. And then this kinase inhibitor, which is a prodrug from Gilead. So again, having access to these motifs now allows their deployment, and you're seeing a reflection of that in the published literature. Finally, the last slide. Let's look at some select emerging bioisosteres, and again, it's hard to select from these. We've seen sulfones mimicking an acid. We use this motif to effect in HIV maturation inhibitors, where we began to view it as a replacement for glycine. Without the burden of a charge. If we had glycine in our molecule with the acid, we had good bioavailability, but not the biology was the virology was not optimal. If we remove the acid, the basic amine gave us potent antiviral properties, but weaker oral bioavailability. This was a nice compromise. Again, we think mimicking the acid of glycine. And then I saw a publication recently from Richard Lee where he had a pantothenic kinase inhibitor where he had a pyridazine in the molecule that was engaging a dual hydrogen bonding interaction with an arginine, just like an acid does. And again, there are very few examples in the literature where this kind of motif has been looked at as an acid isostere. And you can imagine it's complementary to a sulfone. A sulfone is known as an acid isostere. And the geometry is such that the substituents off here come out of the paper or project backwards of the screen orthogonally. This one gives you the planarity that you can sometimes see with glycine. And you would imagine it should be mimicked by a triazole, or even oxidizole or thiazole. And you can restore the geometrical preference by building this around into a ring. Pseudoprolines are interesting motifs with very interesting properties. They've been looked at fairly heavily in the peptide field. There was involvement with Medchem back in the 1970s and angiotensin converting enzyme inhibitors, but these really can be quite synthetically accessible. They have interesting effects on the amide, but really, I think, again, underexploited. There is a large family of these pyrrolidinoazoles and cyclofuse pentaazoles with either the azoles restricted, shown here, or with the nitrogen at the ring junction. This is present, one of these is present in amarigliptin, a long-acting DPP-4 inhibitor from Merck, marketed in Japan as a once-a-week compound. There's a whole raft of bioisosterism you can in these. They've been used here. It's used as a piperazine isostere, and it's been used in glycine transport uptake inhibitors as a piperazine isostere. But I think you could imagine this as a replacement for something like an anisole ring. You've got a nice hybrid of sp2 with sp3 centers and the potential for a little bit of structural inflection, but they generally tend to be flat. So again, somewhat chameleon in nature. The hydrogen bonding effects of a CF2Cl, or the equivalent of that, were highlighted by Frank Beckler. This is Novartis's AVL1 inhibitor. The chlorinated compound is a couple of fold more potent, but that fluorinated compound versus the fluorine, that chlorine engages in a halogen bonding interaction that he identified. And Frank has explored these motifs and shown that these are useful on halogen bond donors and can be useful. They don't suffer obviously from solvation. You've seen also the CF2H attached to an oxygen. This is work from J&J in CR. Sorry, this is work from my colleague Rich Hartz in CRF, where this was worth about 15-fold increase in potency, changing that CF3 to CF2H. And J&J with his factor 11A inhibitors, again, almost a 5-fold increase in potency for changing this from a CH3 to a CHF2, and they showed that that was hydrogen bonding to an element of the protease. These are interesting amino acid, gamma delta amino acid mimics explored extensively by this group in Italy that really haven't found mainstream application, but they've got interesting properties with some nice polarity. Here's the CHNF2, NCHF2 I mentioned earlier. This compound is quite potent when you change that from a methyl or an ethyl to the CHF2. It's equivalent to the ethyl. It's got lower clearance zone. Again, this motif's been extensively explored in the agricultural chemistry field, but we've not explored it anywhere near as intensely. And there's one example in the literature where that change going from a methyl group to CHF2 has a huge impact on drug target residence time, with the CHF2 engaging that receptor much, much longer. The NCF3 is starting to emerge, and here's an example where changing this oxygen to an NH or an NCH3 or an NCF had beneficial properties. And you can see here, this is abrogating, by changing that to NCF3, you're abrogating hERG inhibition, and you're also increasing potency by almost an order of magnitude. And then finally, these aminodioxanes are interesting. Again, these can be viewed as sulfonamide mimetics, maybe a mimic of this thioethene dioxide. Again, I think these have some potential in the drug design. These have been used as mimics of aniline. They won't have the same problems that aniline have with metabolic activation. You've got lone pairs of electrons in here, can mimic the lone pairs in the phenyl ring. And there's a couple of examples of literature where this has been used to replace a phenyl ring and abrogate a hERG issue. So again, I think these are underutilized. We did a perspective on this a couple of years ago on geminal diheteroatomics. So there's fear that these are going to be unraveled in the stomach by hydrolysis. Not if you design them correctly, they're quite stable. And so that's my story. Bioisosteres have a rich history in drug design. I think they're going to continue to do that. And you've seen examples where they can affect potency, PK, and metabolism. And bioisosteres were critical to the discovery of many of these marketed compounds that I talked about. So that I will end. Thank you for listening to me, and I'm happy to engage in questions. Dennis,
Awesome. Yeah.
should I exit screen sharing?
If you like, you can leave it up, but it's
Okay.
maybe if we have to go to a slide, it would be easier to go back. But yeah, thank you
Okay.
so much, Nick. I mean, that was such an amazing tour, and we can see this by the reaction of the audience. I mean, really an amazing tour of so many like even unexpected bioisosteres. I really like that di-THF motif that you showed as
Yeah.
a sulfone isoster, and what you just pointed out on the emerging bioisosteres. Really fascinating stuff. And we do have a lot of audience questions already, so maybe let's directly get started. So
Okay.
here, an anonymous attendee is asking if you can recount any example of how to improve membrane permeability by introducing a fluorine or a chlorine atom without negatively impacting aqueous solubility. Yeah, that's
Well, that's interesting. I mean, clearly, if you introduce a chlorine atom, you will increase lipophilicity more so than a fluorine. The
right.
effect of a fluorine, though, on lipophilicity is modest. The pi coefficient is only about 0.14, so it is a very modest change. Um, I've seen examples where introduction of a single chlorine can dramatically affect metabolic stability. Um, Pfizer has shown that for most developability parameters, membrane permeability, metabolic stability, you can introduce up to 5 fluorine atoms into a molecule without having a sort of deleterious impact, even though that's going to increase the lipophilicity by 0.7. It's difficult to do. I mean, if you want to tie up an intramolecular hydrogen bonding hit an NH and an intramolecular hydrogen bond, and you've got lipophilicity issues, I would go to that to an oxygen atom, which will not have quite the same burden of lipophilicity, although you'll need to carefully regulate or modulate the substituent on that oxygen.
Some, yeah. Yeah, thanks for the perspective. Awesome. Anne Tran here is wondering about deuterium benefits in terms of metabolic stability. She said it can be unpredictable due to the low kinetic isotope effect in vivo in many cases. And she's wondering, in your experience, is there any way to predict when deuterium may have the highest impact during the design stage of molecules?
That's an interesting question. So we actually tried to use deuterium many years ago to interfere with metabolism, and so we installed it at the site of metabolism, and it really didn't have any effect. So I was never likely to start a company like Concert. But what I
Hmm.
didn't realize at the time is that what you have to do is be persistent. So if you look at some of the examples from Concert, we talked to them several years ago about our proteasome for HIV proteasome, atazanavir,
Hmm.
which requires a booster, a cytochrome P450 inhibitor to
Hmm.
give it once a day dosing. They started deuterating that molecule to interfere with the metabolism, and when they blocked the main site of metabolism, it simply went elsewhere. So they were chasing metabolism around the molecule. And I think they ended up with 7 or 8, maybe 9 deuteriums. And you see that in some of their molecules. If you block one site, you simply redirect to another. So it is an active discovery to figure that out. I don't know of a way to predict that. You simply have to do the experiment.
Yeah, totally. Yeah, thank you. Another question here from Manasa. Could you please comment on the reactivity and therefore maybe safety of the CF2CL group, the one that Novartis introduced in that, was it ABL inhibitor?
Yeah, the allosteric modulator. I think in
Right.
that particular, yeah, I don't think there's any problem with them with respect to. I mean, the potential is there for elimination of chlorine. I think Frank did see that on some occasions. It can be a bit of an issue when it's on an ether. If you install it on a benzene ring, a CF2Cl attached to a benzene ring, uh, those will react with a thiol, but only if you heat them to about 150 degrees in DMSO. So those are actually quite stable. It'll be somewhat contextual, no doubt. If you're going to use it on an ether, then I think you would want to de-risk that, yes, by making sure that it is quite stable. But I believe it was quite stable in the context of the Novartis compound. But Frank did see some reactivity, if I remember correctly.
Got it. Yeah, a few attendees here are wondering about the replacement of carbon with boron. Any experience with like bringing in boron into a drug? Of course, that would be probably, reversibly covalent at the least, or maybe even
Dennis,
irreversibly
you were
covalent.
breaking up at the beginning of that question. Can you just repeat it, please?
Oh, sorry about that. The question was about the replacement of carbon with boron.
Carbon with boron. Yeah, okay.
Yeah.
Um, that's interesting. Boronic acids, yes, there are marketed compounds that contain boronic acid, the proteasome inhibitor, and Pfizer have, I think it's an antifungal that contains boronic acid. It still hasn't become, I don't think, mainstream. Um, the proteasome inhibitor is a BOH2
Mhm.
twice full boronic acid. The Pfizer compound is a cyclic one, so it's a monoboronic acid. They have value in certain places, but I don't think you're seeing quite the uptake as some other compounds, other motifs. So again, for me, it would be choosing the right tool for the right target, the right job, if you like. And boronic acid. Um, I did talk about boronic acids and boron as isosteres a few years ago at Drew, and some of my colleagues there were less than enamored with that idea, so I
Mhm.
haven't included it since. And it's probably right; it just hasn't gone mainstream. But again, there's a place where that will be very useful, and certainly in the case of the proteasome inhibitor, it's a little promiscuous, but it seems to work pretty well.
Yeah, no, totally. I've definitely seen drugs containing boron as well.
Hmm. Yeah,
Uh,
yeah.
yeah. So Yoshi is wondering, he has struggled with guanidine isosteres. So
Oh, yeah.
amino heteroarine replacements are as much as he can come up with. Do you have any thoughts on maybe other guanidine isosteres?
Yeah, that's a hard one to replace, depending on the drug target interactions, of course. There is one interesting example which hasn't been published in the mainstream literature from my former colleague Patrick Lamb, who did his PhD with Donald Cram and is a very thinks very carefully and deeply about his molecules. He was working on factor 10A using amides to engage the carboxylic acid down at the bottom of that pocket, the lipophilic pocket, and it forms a tight interaction, dual hydrogen bond. You've got the charge-charge effect. Um, those amidines and guanidines are hard to deliver across membranes because they're always charged, so they don't deprotonate very effectively. You can deliver them by using prodrugs, so that'll at least get them into the body. But if it's an intracellular target, that may be. What he did, he replaced that benzamidine with an iodobenzene, taking advantage of the halogen bonding effect, which he had seen in the crystallographic literature. But this time, this is over 20 years ago, it really hadn't gone mainstream in drug target interactions. And he started out with a 15 picomolar compound and ended up with a molecule with about 15 nanomolar IC50. That's a big change. But when you start in 15 picomolar, 15 nanomolar is still pretty good. And you've got a molecule now you can deliver. So that's the only one I can remember
Mhm.
right off the bat. It has been a bit of a challenge. You can reduce the basicity, and I've seen this done both in thrombin and also in a malaria target using the alkoxy guanidine, where there's an ON bond in there. So you want to install it if it's going to replace arginine, install it in the chain. If you put a hydroxy on the amidine or the guanidine, That's a prodrug. They get reduced down to the guanidine. If you put it in the side chain, that's a lot more stable. It reduces the pKa down to 7 or 8. So now you've got a much better chance for permeability. And I think Merck, along with 3D Pharmaceuticals, took a very I think they advanced one of those molecules into the clinic. So Yoshi, if you send me an email, I'll send you the references.
Awesome. Well, Nick, you're such an encyclopedia. I'm like always impressed how you can pull out of these all of these papers out of your head. That's really, really, really amazing. Yeah, totally appreciate that. We have another question here. What is the probable cause for better properties of CHF2 over trifluoromethyl? So difluoromethyl
Well,
over trifluoromethyl.
yeah, well, obviously it's a little bit less lipophilic, but I think the reason that they created a lot of excitement, over the last sort of 7 or 8 years, was because of the hydrogen bonding potential. But it's not a strong hydrogen bond donor at all. But as you've seen from the protease inhibitor literature, it will engage. Is it, if you look at drug-target interactions, it's an ensemble of all these small interactions that add up to something significant. So the CHF2 can do that.
Mhm.
It clearly works intramolecularly. It's a hydrogen bond donor without the penalty of solvation that you see with a classic NH or an
Yeah.
OH, because it is that bit weaker and it's a bit more lipophilic as well. And so it has been finding application, and as I showed you, it's in two marketed HCV protease inhibitors.
Mhm.
It's there for a reason, and it has its value. But again, like all our bioisosteres, it's going to be contextual. There'll be places where that'll work great. There'll be places where it won't work so great.
Yeah, absolutely. And then another question from Mark Laws is, do you predict a rapid rise in popularity of cubane as a phenyl bioisoster in the same way you showed for the BCPs?
Yeah, that's interesting. I did a review in the Journal of Agricultural and Food Chemistry where I plotted out publications for BCPs and cubanes. Cubanes have been around a lot longer, since mid 1980s. But if you look at the publications, it really hasn't grown very fast.
Mhm.
It has taken off since Craig Williams published or showed in that Angevanta Chemie paper in 2016 that you can replace a phenyl ring with a cubane. And I think he had 6 examples where 5 of them it worked quite well. Um, that has inspired some application, and you've seen publications now learning how to decorate them more effectively. Most recently from David McMillan, looking at substituting in a 1-2 relationship rather than the sort of 1-4 relationship you see in typical cubane inhibitors. Craig went on to make some more analogs and derivatives, penicillin G, for example, and antihistamine, and they ran into a few problems with chemical stability. So whether that's what's reducing cubane uptake, and I don't know. But
Mhm.
certainly the literature is increasing, but it's not to the same level as bicyclopentanes, which have really galvanized the community far more than cubane, despite the fact cubane has been around. Eaton proposed that as a benzene isoster in 1992. And the distance between the vertices is almost identical to benzene. So in that sense, it's actually
Mhm.
better.
Yeah,
But I
yeah.
think, the BCPs are spawning a number of other motifs that are possibly supplanting cubane. And so now, as a medicinal chemist, you've got the choice of probably a dozen of these bicyclic elements with some with heteroatoms in which can really allow you to control the physical chemical properties. The oxobicyclo hexanes, I think it is.
Mhm.
You can install that oxygen atom proximal to an amide moiety and have an NH2O interaction, which will increase the velocity, increase memory and permeability. So again, you've got choices, which is great. And that a
Yeah.
lot of that's coming out of companies like Enamine,
Mhm.
academia, who are driving this bioisostere design. Pavel Mikhailiuk at Enamine has got quite an Alexander Grigenko at the University of Kiev are driving a lot of this with some really interesting small molecules that are structurally quite rigid, but with a dense arrangement of functionality with all of the vectors controlled.
Yeah, awesome. Maybe we have so many more questions, Nick. I don't think we can answer all of them, but we'll end maybe with one of them that also stays on the topic of BCPs. And so the question here would be, many phenols provide pi interactions, maybe, you know, pi alkyl interactions, pi interactions, etc. What would be kind of the equivalent of that in a propylene, you know, interaction?
Yeah. So the pi-pi interactions, yeah, you would assume that you're absolutely require a phenyl ring for that. Mike Bartberger and a guy from Grenadian, I've forgotten his first name, and I apologize for that, explored this 3 or 4 years ago in an ACS Med Chem Let's paper where they specifically looked at the Amgen database, the Merck database, and the DB database of compounds, looking for matched pairs where the phenyl ring was replaced with a cyclohexane, and it had to be a pharmacophore, not a scaffold. And what they ended up concluding was that at about 70% of the time, if you make that change from a phenyl ring to a cyclohexane, you're likely to be within twofold of potency. And they showed examples where you know, where you could replace a cyclohexane ring, a phenyl ring with a cyclohexane in a pi stacking context, and the cyclohexane worked just fine. Again, it will be contextual. There are examples where it doesn't work. So the only trouble with the cyclohexane is you're going to increase livability by half a log unit. Okay, so maybe you could put an oxygen atom or even two in the ring. You know, sugars, glycosides will engage in pi stacking those CH bonds that are axial when the equatorial alkoxys, those CHs are axial, will engage in a CH hydrogen bonding interaction with the phenyl ring, that electron density. So I think there are places to go. And again, you know, a cubane or a BCP might be a little bit too three-dimensional to fit into a stacking site, particularly if it's sandwiched, whereas a phenyl ring would work. You could replace it with an azole or an azine. Those are even better for pi stacking. Again, look at the work of Steve Wheeler and Francois Diederich on pi interactions. They're driven mostly by dipoles. Not always, but mostly. 80% of the variance has been shown to equate with the dipole moment of an azine or an azole. I don't think we think about dipoles enough in drug-target interactions. They can be quite dramatic.
Yeah, yeah. No, well, thank you, Nick. We're over time now. Thank you for a phenomenal presentation, really, and sticking around to answer these audience questions. Again, you're such you know, an encyclopedia of knowledge that, you know, everybody benefits from. We really thank you for, you know, sharing your knowledge with us. And yeah, with that, I want to, you know, thank our audience for the questions. And I will see everybody at the next event, which will be on November 7th. And we'll be talking about drugging GTPases beyond KRAS in the next Flash Talk. All right. With that, have a wonderful day, everyone. And thank you again, Nick. Great presentation.
Thank you, Dennis. Thank you for the opportunity. I appreciate it. Thank you.
All right. Take care.
Good day.
Bye now.